Welcome to the AES Professor's Rounds, Epilepsy Genetics Case Studies. My name is Jill Hunt, Education Coordinator for the AES. Before we get started, I have a few housekeeping notes. This 1-hour interactive session, and we encourage your participation. I will unmute your microphone so you have please keep your microphone muted. You can also use the chat function for questions or comments. The recording of this case conference will be made available on the training continuity section of the AES COVID-19 Resource Library. Your professor today is Dr. Ann Paduri, Director of the Programs in Neurogenetics and Epilepsy Genetics at Boston Children's Hospital and Associate Professor of Neurology at Harvard Medical School. Welcome, Dr. Paduri.
Great. Thanks so much, Jill. And I'm really thankful to the AES for making this opportunity for us to gather available. We are, as everyone knows, in the middle of a global pandemic, and there's a lot of cause for reflection about our society overall these days. And so I think it's particularly important that we take a moment to acknowledge that, but also to acknowledge the great efforts that are being made to keep us together, even if we're physically apart. So with that, I will jump into epilepsy genetics, and we've called this epilepsy genetics case studies, but it's actually a couple of case stories I'd like to share with you with some guiding principles in the middle about how we approach epilepsy genetics cases. And I think, there's certainly time for questions at the end, but if you are wanting me to stop for any clarification or have questions as we're going, just indicate that either in the chat and Jill can unmute you. I won't be able to see the chat while I'm talking, so definitely put something in and Jill's going to be there monitoring to make sure that you can be acknowledged either during or after the slides. So I'm going to go over a general framework for precision medicine and epilepsy because I think that's where these genetics case studies really are key and really important as we're looking forward to where our field is going. I'll go over an epilepsy genetics overview. I'll talk about a precision treatment example. And then there's so much buzz about precision medicine and precision treatment and so on. And so I've somewhat facetiously posed the question of, are we there yet? We always have more to do, but I think we are actually getting there towards precision medicine, and the genetic basis and case-by-case analysis of our patients is how we're going to get there. So here's our first case. And what I would really like to emphasize here is that our precision medicine capability really rests on our precision diagnosis capabilities. And as clinicians, we need to see each case as a potential precision diagnosis opportunity. So this is a little boy named Berg. His parents have allowed me to use his picture and name. And I want you to imagine in your mind healthy parents with their firstborn child who looks like a healthy baby, and then very, very quickly starts to have these stiffening episodes and begins to have what are diagnosed in the local hospital as tonic seizures. So I haven't really given you a lot to sort of reflect on yet, but just think about what you might consider as a differential diagnosis for tonic seizures in the newborn. There should be a whole host of things, not necessarily all genetic, that come to mind. So as time went on, these tonic seizures were refractory to medication, and as we do in child neurology and epilepsy, an EEG was obtained. And it was very, very slow and disorganized and had a periodic quality to it, not completely called burst suppression at the beginning, but it definitely had aspects of suggestion toward burst suppression in the beginning. And there was quite a lot of fear that because he had a disorganized background and multifocal spikes, that he was evolving into something, what we call Ohtahara syndrome, an early infantile epileptic encephalopathy. So this was quite concerning. In addition to the EEG, imaging was done. There was actually a hint of possible cortical malformation in the form of a perisylvian polymicrogyria. And it was actually for that reason that he was referred to me, because I was at the time doing not only epilepsy genetics, but brain malformation genetics. And one of my colleagues said, I really want you to take a look at this scan. And I'll go through all these details because I think it's really important to remember that when we present a finished case, here's the answer, here's the gene, here's the variant. it sort of seems like we knew it all along, but actually, as you all know from your training, there's a long process that goes to your final diagnostic certainty if you can get there. So for this little boy, we actually thought as an infant he might have a cortical malformation, but then we got the images. I really couldn't find it. I called my colleagues in radiology, who I rely on very closely, and said, I'm not going to tell you where somebody thought there was something, but what do you think? And then we looked at it very carefully together, and we could not convince ourselves that the MRI was abnormal. We repeated it again post-infancy and confirmed our suspicions that it had just been maybe a funny cut or something. So here's a child now with a normal MRI, early onset, bad seizures, abnormal EEG, epileptic encephalopathy. And this was several years ago, back in the 2011-2012 era, where we were just beginning to do epilepsy panels and things like that, and where we had just seen some publications suggesting what some early onset epilepsy genes might be. So think in your minds what some of the genes you might consider would be, but also remind yourself that when I was in this place, we only had a couple of genes to think about because we didn't have this whole long list of genes we know about today. So in this case, Med1 was used, and then Med2 was used, and then Med3 was used. And I'm intentionally not telling you what they were because actually they're not related in any way to anything specific about what the child had. And eventually, within the first year of life, the seizures subsided. And actually, developmentally, he didn't do quite as terribly as we thought he might. Developmentally, he was beginning to start to walk. Shortly after a year of age, he actually was smiling and very alert and looked quite engaged. And so we were quite hopeful that he might do okay. But in fact, he continues now several years later, still smiling, still walking, but nonverbal. And so he certainly does have the consequences of an early epileptic encephalopathy. So what it required really to get to that point in our understanding of him was first to sort of see what the positive points were in our data and also to exclude things that were irrelevant. And then really focus on the idea that we had an unexplained tonic seizures, epileptic encephalopathy during infancy. Unexplained epilepsy today should really trigger in your minds, it's probably genetic. So what really has to happen is that all of our years of genomic science should really help us with our precision diagnosis. And only then when we combine these aspects can we have precision care. So I want you to have Berg in your minds. And when I get to the gene that's his gene that explains everything, even though he stopped having seizures before we got to that genetic diagnosis, you'll remember this story hopefully, and we can talk a little bit more about him as we go through. So the goal really is to combine all those aspects though and have precision medicine for our patients with epilepsy. I'm just going to go through a schema. This is really how I view every case and how every case in my mind interacts with this inner circle here of my clinical world, and then I'm going to build an outer circle for you to talk about how I think all of these things are connected. And so, Tuesday afternoons, I'm typically in my clinic, and I'll make a diagnosis of epilepsy, recurrent unprovoked seizures, as you all know. And it's important to sometimes, even among ourselves, among epilepsy-inclined people, to remind ourselves of that definition, because I'll get sent patients with a variant in such and such SCN1A, for example, gene that's an epilepsy gene. But if the patient doesn't have epilepsy, it really changes how I'm going to interpret the variant in that particular patient's context. So remember, go through the basics, even if you're in a super, super specialized clinic. Remind yourselves, look at what the history is, see what the EEG showed, make the diagnosis of epilepsy. So we do that. We now can undertake a genetic evaluation in a pretty straightforward, routine way for at least some cases and some insurance plans and some settings, but not everybody can, so it's important to keep that in mind. And then we pursue clinical treatment, and our clinical treatment today is largely just empiric treatment, the same as it was when I was a fellow and the same as it was when my attendings were fellows, because we really haven't pushed our genetic knowledge yet into routine changes in clinical management for our patients. And I think that really should be the goal. We have all of this accumulated science, all this amazing understanding of the epilepsy syndromes, and all of that could be put together, hopefully to make a dent in how we do for treatment. Because as you all know, at least a third of our patients don't do well with the treatments that otherwise should rationally work. And in these early onset pediatric patients, they do even worse. And so patients who have epileptic encephalopathy in the acute period typically have refractory epilepsy. Some, like Berg, don't necessarily continue with those seizures, but that's after weeks, if not months, of unrelenting seizures. So how are we going to change this? We're going to build this outer circle where we have registries and natural histories, which might be based on syndromes, like Ohtahara syndrome, for example. Eventually, they might be based more on genes. So KCNQ2, that's what Berg had as a gene explaining his condition. He had a pathogenic variant in KCNQ2. And we built a natural history study, a pilot study now around KCNQ2 with some colleagues and some other genes. And we contribute to many other registries and natural history studies. And that's a sort of hub and spoke model that I hope everybody can adopt in your careers. Or maybe you'll be the center of attention and research focus and clinical focus on a gene or two if you're going to be doing genetic epilepsy, but then you'll have your other 98% of your patients. And if you can find a way to connect those patients to the natural history studies and registries of your colleagues, then that hopefully will be a productive way to get all of our patients engaged in some form of research that will help drive our knowledge about what these conditions look like. The gene discovery is continuing, so I spend most of my time thinking about gene discovery, enrolling patients, looking at phenotypes, and then trying to push the envelope in terms of what known genes can do and what new genes might be part of the story of epilepsy. And so that's a big part of what needs to be brought into the clinic in the form of translation of panels, microarrays, exome sequencing, eventually genome sequencing. Once you get a report, which any of you have seen a report will know, you get a report, you may get a list of genes and a list of nucleotides and amino acids, and there's an interpretation on that report. And what goes into that interpretation is a whole science unto itself. And that's important to recognize that there's a whole set of criteria that we use to judge whether a variant may or may not be associated with a given condition. But a lot of times we don't have all the information we need, so you end up with a lot of variants of uncertain significance, for example. So important to keep that in mind. Once we have a new gene or new variants in a gene, we do need models, whether they're cellular models or animal models, to be able to assess their pathogenicity. And then really before we can have precision treatment based on these genes and variants, we need preclinical data, preclinical trials to inform precision management. So that's a lot to ask. It's more than any one group can do, and it really calls for a collaborative environment where we share our patient data across institutions, where we share gene discoveries, where we get clinical labs to adopt new gene discoveries and offer them for patients, and where we challenge our institutions where coverage for these tests may not be offered routinely to try to offer them. And that way we hope we can get to the point that we're no longer 10 years from now saying that a third of patients are refractory to their medication, but rather if you have a patient and you have a specific cause, this is how many will be doing well. So I hope in 10 years when one of you is giving this lecture, things will be a little bit brighter of an outlook in terms of our treatment. So with that, I'm going to focus the rest of this on gene discovery and talk about how we can contextualize all of the various discoveries that are coming out of the field. And I'll pepper the rest of this with some stories, and I'll end with a story where we actually have been able to use precision treatment. So just by way of overview, predating all of us in the field back in the 70s, but even before this, was this concept that we had a few known causes for epilepsy: trauma, stroke, congenital lesions, HIE, and so on, but the vast majority of the cases were considered idiopathic, which then evolved by the time I was in training to be called presumed genetic. We sort of figured if you're not able to come up with an acquired cause, and they had non-acquired epilepsy, and that was most likely going to be genetic. And there's a lot of data supporting that. Twin studies where monozygotic have more of a rate of concordance than dizygotic twins, family studies where multiple people in multiple generations are affected. So a lot of data suggesting that these presumed genetic cases really did have a genetic basis. Fast forward to now, where yes, we have more imaging for sure than in the 70s. And so we have a certain number of cases who have focal lesions that are detectable and genetic in origin as well. We've recognized the autoimmune encephalitides that can have epilepsy as a component, which is really a growing area where I think we're going to learn more and more about those. But we've solved part of this big piece of the puzzle about genetic epilepsies, mostly with single genes. And now we're sort of at this kind of leading edge of trying to figure out what some modifier genes might be, and we're looking at non-coding regions of DNA and so on. So we've definitely gotten to the point of proof of concept, and we've gotten some really tractable, important information for our patients, but we still have a lot to learn. So just to give you an idea of some of the genes that have come out, you'll have heard of some of these. KCNQ2, which is the one that explains Berg's condition, the Oda-Hara syndrome, or the tonic seizures of infancy with a disorganized EEG, typically burst suppression. That's one of the first epilepsy genes that was known. And I'll just give you my own historical context. Back here is when I was in medical medical school, and there was just this one gene. And by the time I graduated, this second gene. And then fast forward to when I finished my epilepsy fellowship, there were a handful of others. And to go from one to, half a dozen was already a lot of progress. But then we had a little bit of a lull. Everybody was looking for genome-wide associations. And there's not just one epilepsy, as those diagrams imply that epilepsies are a group of conditions where epilepsy is a prominent feature. Putting all of those different types of epilepsies together and looking for genetic signal didn't work out so well. So our era of GWAS was not all that successful in the early days. But what has been successful is the last 10 or so years of next generation sequencing, where instead of looking at one gene at a time, one base pair at a time, we can look across all of the genes. We can look at panels of dozens of genes, or we can look at the whole exome, and increasingly we can look at the whole genome. And that has allowed us to identify variants in a whole host of other genes that are quite important. And so, whereas in the sort of mid 2000s, we were hearing about PCDH19, what was called epilepsy in females with mental retardation initially, and now called girls clustering epilepsy. It was a Dravet-like presentation with fever at the onset, but only in girls, and sometimes inheriting variants from their unaffected fathers, which threw everybody off because that's not usually how genetics works. These cases were really important and interesting, but they came to be because of a collection of cases and a collection of families that really made the case from a genetic perspective that this gene was important. And it's only now that we've been able to validate that with large population studies. So our beginnings here back in these early days with SCN2A, SCN1A, KCNQ2, SCN, SLC2A1, all of these relied on really deep phenotyping and big family studies, some studies of individual cases being brought together, and large-scale collaborations between clinicians and clinician researchers and bench scientists to be able to make these discoveries. The same is true today. The difference is that the scale of the number of genetic variants that we're coming out with is huge. It still requires careful phenotyping to understand what they mean. And it still requires basic knowledge of what these genes do. And so the whole concept hasn't changed, but the scale has changed dramatically. And because of this massive volume now of epilepsy-related genes, it really does require a team effort across institutions and shared platforms where we can actually think about our genetic variants together and think about a logical way to analyze the variants. So I'll just pause for a second to mention that the ClinGen initiative through the NICHD, which is largely being led now by Heather Mefford and Ingo Helbig with respect to the epilepsy genes, is a way that any of you, if you're interested, can come together to ask questions about variants in a given gene, but more importantly, get involved in the curation and get involved when it's time for DNM1 to be curated or to be recurated. Take a crack at one of these genes and take a crack at looking at the literature and the variants and presenting to a group of people like me who will be able to give feedback and be able to comment on our experiences from the dark ages of epilepsy genetics into the modern era. So all of these things are more than any of us can keep in our minds and be aware of in a fluent way immediately. It really does require this group approach to be able to intelligently work through these and bring them to our patients. So as I was alluding to, there's lots and lots of genes. If you look up epilepsy in PubMed, you get several hundreds of genes, and there are new genes coming out for many syndromes. There's new phenotypes with known genes being published all the time. How do we make sense of this and bring it to our patients? So one way we can do that is by parsing out our epilepsy by syndrome. So this is good old-fashioned epilepsy phenotyping, epilepsy classification. We have our International League Against Epilepsy vocabulary that we can use, and it's a shared lexicon, so we can actually talk about things using the same language. And so granted, it changes over time, reflecting the improving and sometimes changing concepts that we have, but what was partial epilepsy became localization-related epilepsy and then went back to now being focal epilepsy. But if I now say somebody has non-acquired focal epilepsy, that tells you something. That tells you that they have a focus on their EEG, most likely, or a semiology suggesting focal epilepsy. And it tells you that they're non-acquired, meaning imaging has been done in 2020. There's no obvious structural cause. There's nothing in the history suggesting a remote cause. And so it's suspected genetic. So the words that we use to classify our patients on an individual basis are very helpful patient by patient. They're also really helpful for creating a hypothesis in our minds about what type of genes might be involved, because there is some difference syndrome to syndrome. So I'll just give you some examples. We talked about Berg and his tonic seizures evoking at least a seizure type of Ohtahara syndrome, even though he didn't have the full seizure, the full EEG presentation to go along with it. And he had a variant in KCNQ2. It turns out if you look at cohorts of patients with the Ohtahara syndrome, which we had the opportunity to do at Boston Children's with Heather Olson, who was a fellow and is now attending in our practice, what we found is that you could actually figure out over half of them just by using exome sequencing and honing in on a handful of genes. KCNQ2 was the top one. Close second was SCN2A. And then there are several other genes, STXBP1, and some others that showed up on the list. But what that tells you is if you see a baby in the NICU, let's say, or consulted to the NICU, and they have what looks like it might be evolving into Ohtahara syndrome, you should be thinking KCNQ2, 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 SCN2A, SCN2A, handful of other things. Gee, a panel where I can saturate those genes and get sequencing with every base pair covered and get deletion and duplication testing of all of those particular genes that are known to be associated with epilepsy today in 2020, that's where I'm going to start. And if I can get that quickly, great. If I can get an exome more quickly, also great. But remember that the exome is not going to tell you if you've got a, let's say, whole exon missing from one of those genes. So you want to make sure you cover all your bases with the testing if you've got a strong hypothesis. So let's say I choose an epilepsy panel to do and I get the result that the panel's negative. Does that mean that they don't have KCNQ2 epilepsy? Well, it depends whether KCNQ2 is on the panel or not. I mentioned that was an old gene, so yes, of course it is, but new genes are coming out all the time. So if you see a patient and they've got a specific syndrome, and you've got that gene in your mind and you're sending a panel, make sure that that gene is on that panel. Similarly, this is a sort of pet area of mine clinically and on a research basis, epilepsy of infancy with migrating focal seizures. There we have some overlap, but actually the top gene is KCNT1, a different channel encoding gene. And this is one that explains maybe about a quarter to a third of all of the babies with epilepsy of infancy with migrating focal seizures. What other genes are involved? Again, there's some overlap. So there are some KCNQ2, some SCN2A, and some others, actually a whole long list of other genes that we've identified. So that's important as well. And in this case, if I were doing testing in a baby, I'd probably start with either a panel that definitely had KCNT1 on it, as well as these other two genes, or possibly exome sequencing, because we've found de novo, we've had an inherited, we found recessive, we found all sorts of mechanisms giving rise to the genetic causes of epilepsy of infancy with migrating focal seizures. This is a condition that was described when I was in medical school in the 90s. It was pronounced to be non-genetic in the early 2000s because all epilepsy genes had been sequenced and tested and they were negative. All three of them. And there weren't really sophisticated ways of doing the testing efficiently then. And so now that we've had bigger cohorts to look at, we've been able to look at larger groups of patients, so over 120 patients, and we've been able to look at all of the genes. We've actually been able to paint the genetic landscape of this syndrome that for many years was considered not to be genetic. So this is really a nice example of bringing in that clinical phenotyping, bringing in that classification that we all learn about in fellowship, and then drawing the genetics into that. These are all relatively rare syndromes, as you know, but infantile spasms is less rare. It's the most common of these syndromes, these early onset syndromes, and it's present in about 1 in 2,000 babies. So that's about as common as Down syndrome, so not uncommon. I'm sure if you took a poll of general pediatricians and asked them how many patients that they have with Down syndrome, they'd be able to think in their mind of a few of them. But if you asked, how many kids do you have in your general pediatric practice with infantile spasms, they may not know unless they've just had one recently. And I point this out because babies with infantile spasms often grow up to be kids with ongoing epilepsy, sometimes Lennox-Gastaut syndrome, often with intellectual disability, the majority with intellectual disability. And so the pediatrician may not necessarily recognize that way back when this child was a baby with infantile spasms. That's important because we as epileptologists will take that history. We'll ask the parents what the seizures look like. We'll look for the EEG data. And it's important because we've got a long list now of genes accounting for infantile spasms. And so in the non-syndromic baby with a normal MRI and no HIE as a clear explanation, you're thinking genetic, and there's a long list of genetic possibilities. So again, here, a panel with a lot of epilepsy genes would be a reasonable place to start, but an exome would not be an unreasonable place to start either. So I've been throwing some gene names out. Of course, there are many, many more than I've mentioned. This is where the font gets small enough that I've got to put the glasses on, because you can see, if I look back at these syndromes that I mentioned, I talked about the early onset epileptic encephalopathies with KCNQ2, like Berg, the little boy from the beginning. I talked about EIMFS, where KCNT1 is a tried and true one, but we've got several others, and this is already out of date now, a few years in, but this is a nice review looking at these. If you look at infantile spasms, again, lots and lots of genes, more than we can list, but you can see there's an overlap between the early onset epilepsy and infantile encephalopathy and the infantile spasms group. So what does this tell us? It tells us there's a lot of genes, there's a lot of overlap on the gene lists, but that some conditions really do have predominant genes. So this one, KCNT1 is important. Dravet syndrome, I'm sure you all know, with severe myoclonic epilepsy of infancy. SCN1A is the top 9 of 10 genes if you've got a case with what sounds like Dravet syndrome, fever at the onset, hemiconvulsive seizure, developmental plateau in some cases, and decline and evolution of the EEG to involve generalized seizures, and then myoclonus developing. That's Dravet syndrome, that's SCN1A until proven otherwise. You're really going to saturate that gene, versus a condition like infantile spasms where you may want to look at lots and lots of genes. Eventually, though, I hope we can move away from the syndromic classification, which is a really good way for us as clinicians at the bedside to formulate a hypothesis. I hope we can move from there to a neurobiological characterization, because ultimately our treatments are not necessarily going to respect the ILE classifications. That's how we see them, that's how we can conceptualize them, and that's how we can categorize our patients and form hypotheses. But as you saw, there's a lot of overlap between the genes that are in these different conditions, and ultimately what's going to drive precision treatment and a scientifically based treatment is going to be the neurobiology. So we're going to have genes that are expressed at the synaptic cleft, we're going to have genes that are expressed in the reuptake pumps and the vesicles, we're going to have genes that are expressed in the axons, and all of these different places and proteins and functions are going to be susceptible to modifications with drugs and potentially with gene-based therapies. And so probably where we're going to classify these patients is based on these neurobiological categories. We just don't have enough data yet. So moving from the past to the present, where we've got all of these different options and we at least have a working way to clinically conceptualize syndromes and a working hypotheses for each of these different types of syndromes, what explains the unsolved? So I've told you that in some of these syndromes, up to half or even more of them, the kids can be explained. The EIMFS, where at about 2/3 can be explained. The Ohtahara syndrome, half to 2/3 can be explained. Similar for infantile spasms, about 40 to 50% in most of the series. So that's a lot. That's a whole lot more than the 0 where we started when I was in your shoes. And so I think it's fabulous that in the last 15 years we have all this knowledge, but it still means we've got lots and lots of patients with non-acquired epilepsy, often with accompanying encephalopathy and intellectual disability, with a normal MRI, who still have unexplained epilepsy. And I would challenge you to continue to ask yourself with those patients, what do they have? And if they have, let's say, Ohtahara syndrome, and they look like they're having tonic seizures, and it looks like it's got to be one of those genes, and your first pass testing is negative, look harder. Maybe they've got something that's in a non-coding region around one of those genes. Maybe they have something in a gene related to one of those genes that hasn't been discovered yet. And that sort of knowledge base keeps growing, but they're going to be continuing cases that are unsolved and unknown, and we have to just not give up on them as well. So one reason we may not find some of the variants is because they may be mosaic. So when I say that Berg had a KCNQ2 variant that was pathogenic, it was a heterozygous variant, meaning one of his 2 copies of KCNQ2 had a variant in it. And that was present in every cell in his body. So when we do DNA testing, what are we usually testing? Usually we're taking a venipuncture, we're taking a blood sample, and the white blood cells, which have nuclei that we can get DNA from, are going to be the source of our DNA, and we're going to test that white blood cell DNA. And if all of your cells have a variant in it, you're going to find it in that white blood cell DNA. Even easier today, and particularly given the current circumstances where people are not necessarily inclined to go to testing labs if they don't have to, we can take a cheek swab, and you can do a cheek swab assessment, get the cells from the side of the mouth, from the epithelium, and those are a source of DNA that can be tested. And so there's many ways to get DNA non-invasively or very minimally invasively with venipuncture that we usually use. But let's say you don't have necessarily heterozygous variant in KCNQ2 present in all of your cells. Let's say when you were being formed a few cell divisions in, that's when the mutational event occurred, and you've got a subset of your cells containing a variant, then it may not be possible to detect that using current detection methods. It may be that, let's say 20% of all of your cells, brain and blood and everywhere, has it. So that means that the tissue source of blood may still be a reasonable tissue source. It's just that our analysis isn't tuned in to pick those up yet in a sensitive way in most clinical labs. But it may be that it was a change that's affecting just the brain tissue. And so unless you had brain tissue, you're not going to find it, and you're not going to biopsy all of your negative patients to see if they might have a mosaic variant, particularly when I've just told you they can be hard to detect. So where we have looked at this most actively and where we've made the most progress is actually not so much in these non-acquired epilepsies and the early onset epileptic encephalopathies, but actually in patients with malformations. So I'll walk you through this because it illustrates the concept, but really it's a way to say that I think the mosaicism is there in other cases. We just haven't been able to sensitively identify it yet. So this is an example of a large brain malformation, hemimegalencephaly, where because of an early mutation in development, let's say a third of the cells in early development are affected, and those 1/3 of the cells spread out across the brain. They happen to be on one side of the brain, and this child has hemimegalencephaly with some fraction of their brain cells enlarged and carrying the variants. And so to access that tissue through epilepsy surgery is something that used to happen more routinely. Now these kids are having more of a functional disconnection. But when you take that tissue and you look at the changes in that brain, you look at the DNA and you compare it, let's say, to a buccal swab source or a venipuncture source, you can do a same different comparison and say, oh, look, there's a variant in this brain tissue DNA that's not everywhere else. That's what we call a somatic mutation or a post-zygotic mutation that leads to mosaic variants. So that was a little bit yesterday's news already. We were much more excited because hemimeg is big and it's terrible and devastating, but we only see it maybe once or twice a year in a large referral center. But if you then walk the concept back to something smaller like focal cortical dysplasia, that's something that you see all the time in the epilepsy practice. Many of the old cases of non-acquired focal epilepsy that we thought were non-lesional, meaning the MRI looked normal, now on re-imaging are showing up with small lesions suggesting a focal cortical dysplasia. But under the microscope, the cell type looks the same as with the hemimeg, and that told us something, and it told us that there's probably post-zygotic mutation and mosaic variants going on there, but they're going to be in smaller number. You've got less number of cells affected. You've got basically a little patchwork quilt of just a few mutation-bearing cells. And so that's a little bit harder to detect, but it has been detectable. And so for this condition, hemimeg that we see again once or twice a year, about 2/3 of the cases now can be solved, even clinically, although it's not done routinely, but there are a couple of labs in the US at least that will do testing. These are mostly variants in the mTOR pathway. And so we'd like to be able to do this a little bit more quickly and aggressively in terms of the testing. And maybe it's not going to require brain tissue. Maybe we'll be able to do this with spinal fluid if there's cell-free DNA floating around. And so colleagues of ours in Australia and other places are looking into this as a possibility because it really could be game-changing to tell the family of a child with hemimegalencephaly, let's say you've got a variant in the AKT3 gene, it's revving up mTOR, and we have mTOR inhibitors we can apply. Why don't we try that before hemispherectomy? That's the goal there. But same concept, and in many cases the same genes with focal cortical dysplasia that we all see all the time in epilepsy practice. And so here we've been able to solve in our cohorts at Boston Children's, colleagues in France and colleagues in Korea and several other centers, about 40% of the cases now have one of two types of variants, either the same type of mTOR variants that these large lesions taught us about in a variety of genes in the mTOR pathway, or in another gene called SLC35A2, which is involved in glycosylation. It's actually a congenital disorder of glycosylation gene by its history, but when it's present in the brain, it's X-linked, but it can be present in the brain in this post-zygotic patchwork quilt in boys or in girls. It can give you an FCD. It looks a little bit different than the classic type 2 FCDs that you're used to seeing in most of the patients you see, but it gives you an FCD-like pattern. So all of a sudden we went from having 0 answers to 2/3 of these cases solved in less than 10 years. And really over the last 5 years, almost none of these solvable to now in a research setting about 40%. So where am I going with this and how do we take this back to those other cases? Well, suppose you have a brain that looks normal, but suppose, not late, but still pretty early, but later than in these cases, you had a postzygotic mutation, a spontaneous mutation in a gene. It could be in the same pathway in mTOR, it could be in that CDG pathway, or it could be in KCNQ2 or SCN2A or any epilepsy-related gene. That set of cells, a small number of cells, are going to be then giving rise to a smaller area of abnormal cells, let's say, for example, in the hippocampus here. And so it's going to be very hard to detect that even if you had brain tissue, but it could give you a localized and potentially only microscopically visible epileptic network where you've got, let's say, 2% of the cells with an SCN1A variant disrupting the electrical stability of that region. So this is where I think one of the major unsolved areas is, and one where I hope we'll make progress in the coming decade. Why else are we maybe not solving all of our cases? I'll just go through this quickly. It's something that we're beginning to look at, but when I talk about KCNQ2 and SCN2A and all these genes and interrogating them with panels and sequencing with exome, I'm talking about looking at the coding sequence. These exons and introns look like this, and we were really only looking at the exons, and the entire exonic content of our DNA is only about 1% of our DNA. So we're looking at a lot, we're making good progress, but we're ignoring 99% of the DNA. So what's in there? There are all sorts of things that are upstream and downstream of our gene. So imagine here there's something that's telling gene 2 to turn itself on in certain tissues, and then there are areas called repressors which turn genes off, and in every different tissue type these may be expressed at different levels at different times during development. So it's a rich and complicated system, but it also is not one that we've had a lot of experience interrogating clinically. So that's something that's, I think, on the horizon. And then finally, what explains a lot of the unsolved cases is we're not testing them all. So I've told you that we can. As you can tell, I'm very excited about the science, but we're not always making it over to the clinic to get the testing done for our patients. And that's due to a number of different factors. I have the privilege to work with an international group where we're trying to come up with some general guidelines for what types of patients should be tested. And this is driven in large part by Lynette Sadler in New Zealand, who's kind of leading this effort to say, well, let's put out there what we think any child with an developmental and epileptic encephalopathy should have as a genetic evaluation. And then let's talk about how those various tests we recommend may or may not be available in every setting. So from a global perspective, there are many places where there just simply aren't the resources yet today to be able to do the genetic testing. Some of that will change because the cost of genetic testing is going to continue to go down. That is happening. So hopefully that will change. And some of that hopefully will change as more treatments become available. It's going to become an imperative to say, look, if I test 100 babies and 50 of them will have an answer and 10 of them I might be able to give a precise treatment to, that's a start. I hope that number goes up, and I think that's going to help us drive our systems wherever we are and wherever we're practicing to really push for more testing and earlier testing, particularly for the young patients who have refractory epilepsy, and particularly for the ones with comorbid intellectual disability, because the yield there is very high. The other reason the patients aren't tested, though, even in places where there are resources, is that depending on the healthcare system you're in, there may be no testing allowed, all testing allowed, or as it is in the US, it depends. And it depends on your insurance, and it depends on all sorts of things. And it's several people's full-time job to figure this out. And it's more than we as physicians can usually figure out. So I've partnered not only with a team of genetic counselors who really help us navigate the system, but also now with our departmental leadership and hospital leadership to try to navigate a way to be able to get every patient tested whom we really think needs to be tested. And one way that we're trying to demonstrate that that's the case is through an institutional pilot study. And what we're doing there is we've been given permission over a couple of years to enroll 500 children with epilepsy that's unexplained, and their parents, and to do trio sequencing. And the idea of this is to get diagnoses for individual patients who otherwise might not be able to do so, but also to be able to show what the outcomes are. And those outcomes might be change in treatment. The outcomes might also be, we found a variant in a gene that tells us that the person needs to be screened for kidney lesions or other forms of disease that might otherwise not be thought about. So we do this sequencing. We actually have a commercial company doing the sequencing, sending us the data. We manually create these variants, and we look at the gene level evidence, the variant level evidence, and then we can give results to patients and their physicians who are treating them, along with, if requested, treatment recommendations if they exist. So that's about 1 in 10 of the cases that we've seen so far ends up in this category. The others, we're still kind of working through the analysis and still enrolling patients, but the other ones, we need further evidence. So we're looking for collaborators who are interested in the same gene. There's a program called Gene Matcher where you can put your case into a database, and then if anytime somebody has a case with the same gene involved, you can be queried with your permission and mutual permission. So there's all sorts of ways to try to connect globally to try to put cases together. And then finally, we do need experimental models. So my lab is working on zebrafish models. My colleagues are working on mice. Other colleagues are working on induced pluripotent stem cells that are turned into neurons and are corrected for just the variant a patient may have. So there's lots of different ways to look at this. But this needs to be done if we're going to be able to make more sense of more of our patients. So I think this today is, of the unsolved cases, the most tractable and the most solvable. We don't need more science to do this. We need more patients to be sequenced. So just to give you an idea of some of the genes that we're finding, we've actually analyzed now over 300 cases. We've found variants in genes like SCN1A, which I've mentioned, like KCNQ2, whose protein product binds to KCNQ3, and lots of other genes, some of which may be familiar to you, some of which may not. And so we have patients who have variants who have had the features. So SHANK3, for example, is a gene associated with Phelan-McDermott syndrome with intellectual disability, predominant, and epilepsy. And we saw a young woman in her 20s brought in by her parents. And all over the notes, it was suspected that she had this, but due to systemic issues, she wasn't able to get testing. Because of that, there's not a huge concept change for her providers about what she has, and her seizures are actually relatively well controlled. So you might say, why does that make a difference? But for that young woman, the parents, first of all, have an answer after 23 years. They're not blaming themselves for something that they did or didn't do during pregnancy, or something that may have helped her that they didn't provide for her. They now have an answer about what happened. They also have a community. They have a community of other parents. It so happens that there's an NIH natural history study, so now they can contribute their child's data to be able to help other families understand the condition, to look at natural history, and potentially to participate in a trial if such a trial comes about and she's eligible. And so just the diagnosis alone has opened all sorts of doors for that family. And we're counting that as outcome as well. So I think this is just one area where I think we can make a huge difference just by asking the question, why? Why does this patient have epilepsy? And maybe to you that's sort of obvious, but I do try to mention that in these lectures because when I was in training, we didn't have a lot of tools to ask why. But the natural course of the day was you'd see a patient, you'd sort of treat them empirically, and you'd kind of move on. And this, let's spend a few minutes thinking about why, wasn't part of what we did. And because of the genomic revolution, we really have to sort of change our paradigm. We have these sort of set ways that we do things. We take our history in a certain order. At this point, we might want to bulk up the family history section a little bit more. We do our exam, potentially with specific conditions in mind. But now when we get a genetic variant, it might be time to go back and say, the kids with TCF4 variants, they usually have a particular look to their face. Does my patient have that? Let me go back and look. And you may have to do what I call recursive phenotyping, where you get a genetic explanation, you're not sure if it fits. You've got to go back and bring the child back and take a look at them. And in some settings, that's going to be done by geneticists. In other settings, it's going to be done by us, neurologists with a genetic bent or postdoctoral training in neurogenetics, or possibly the few of us in the field, not myself not included, but the few of our colleagues who've actually gone about dual training in neurology and genetics. So I think there's lots of different ways to approach this, but it starts with asking why. You'll see here in this blue box lots of genes where we don't really know whether they're specifically epilepsy-related genes or if they're genes where they're variants that are along for the ride. So this sort of represents another several thousand person years of work to figure that out. For some of these patients, though, just even in the last year that we've been doing through this research / clinical project, we've actually been able to implement considerations for treatment for a number of these genes. We have direct implications for recurrence risk when they're recessive or X-linked. And even for de novo, we've now learned that we should tell parents that you may have another child with this condition because of mosaicism in the parents. So the parents' oocytes or spermatocytes may well be mosaic for a particular variant, which means the second child actually could, in theory, have the same variant. And so just finding it de novo doesn't mean no chance of recurrence, which is also really important. So that's a lot of background. This was supposed to be a case series. I've tried to sprinkle in some cases as I went along, but I want to spend a few minutes on one of the cases that has really touched me, partly because of how we've been able to make a difference, but also because it's been really, really simple, but it required a lot of people asking why. So this is a little girl who presented when she was about 3 years old with epilepsy, with generalized epilepsy, mixed generalized epilepsy with generalized tonic-clonic seizures, with tonic seizures and atonic seizures. And in addition to the seizures, she became very hypotonic. She became rather ataxic and had difficulty walking independently, which she had been doing before. And she stopped saying the few words that she'd been saying. So this is the child, and you can see here she has a brother, and what they have is actually two variants in the same gene called CAD or CAD. I'll just pause for a second to say that that's not why they came to see me in our epilepsy genetics program. They actually came to see me because they both were found to have a variant in a gene called GRIN2A, which is actually on some of those lists I've showed you. But it turns out that that was inherited from a healthy mom. And while some genes can give you that scenario, this is one that typically doesn't when we're talking about a pretty severe epileptic encephalopathy here. And so the geneticist who was seeing her, poor old Jayakar in Florida, said, you know, I don't buy that, I'm going to go further, and did an exome sequence. And by the time exome sequencing came back, we were seeing each other. And they have these two variants, one from mom and one from dad, in this gene called CAD. What that has been associated with, we now know, is atrophy of the cerebellum. And so this is what the brother had. This is what the sister had. I'll blow up hers a little bit. So this is the child we saw. So I want to just take a second here to talk about diagnostic precision again, because initially this child's MRI had been read as normal, and we got the imaging and we looked and said, you really shouldn't see cerebellar folia that prominent. This looks like generalized seizures, hypotonia, cerebellar atrophy. I know I can't hear you, but I just want you to think for a second what that sounds like to you. To me, it sounds like a congenital disorder of glycosylation. That's what I thought it was. I sent off serum for transferrin isoelectric focusing to look for which CDG this was. Because this CAD gene that had these two variants hadn't been described yet. Within the next few weeks, a paper came out, and then a footnote came out in a metabolic textbook that my colleague Jerry Berry down the hall from me had, that there's a new publication from Austria with a handful of kids with two variants in this gene called CAD. So what does this do? It's a multi-enzyme complex that's involved in pyrimidine synthesis. So we have our purines and our pyrimidines, and the pyrimidines are not synthesized normally if this is defective, but we do have a salvage pathway, and it stood to reason that maybe you could rectify it by giving it. You can't give back your T of your ATCG, but you can give back U. You can give uridine, which is a substrate for your pyrimidines, and that is able to be used by the cells. And so this sounded good in theory. A couple of the kids in the Australian series had had exposure to this. We thought we would do some zebrafish experiments because we saw that this is just a heat map of the motor. We saw in the first generations of fish injected with CAD, knocking it out, CAD constructs to knock it out, lots and lots of seizure-like behavior in the fish as opposed to the wild type. And so we were going to go testing them for uridine improvement, but unfortunately they were dying off too fast and we couldn't really get our experiments done to completion. But then this Austrian paper came out, and we said, well, why don't we just try them, use the doses that they used of uridine. If she can't make it herself, let's give it back. And it's something you can buy at the vitamin store. We were very comfortable trying this, and we also saw in her blood smear these abnormal cells in a funny pattern called anisopoikilocytosis, which is basically abnormally shaped and sized red cells. We showed our hematologist, and they said, yeah, this looks like a blood dyscrasia. And in fact, it is, and that's also described in the original report. So we said, well, let's see how she does. We're going to follow her seizures. We're going to follow her development. We're going to follow her MRI. And now we've got another biomarker. We're going to follow her blood smear. So a year later, this is how she looked. And I promise this isn't a bad day in 2017 and a good day in 2018. She looked a lot better. Her tone was back. She was able to walk independently. She started smiling and interacting. But she still had those weird cells on her smear. So we said, well, that's, you know, one of our biomarkers is not looking good. Let's go up. So we went up on the dose again. And here she is last spring in 2019. Hopefully this will show. We never thought she was going to be doing anything like this when we first saw her. We were really hoping to stabilize her, but she's actually able to run and climb and do all sorts of things independently.
Say hi. Okay.
And now she's quite a firecracker. I saw her in a virtual clinic a couple of weeks ago, and she's able to say full sentences and tell her mom she wants to leave and all sorts of things. This was actually last year when she came to clinic and swiped our genetic counselor Sonal's sweatshirt from her. But she's really made tremendous progress. She's not normal, but she's functioning probably at a preschool level, and she's at the first grade age level. So not perfect, but substantial improvement every year with an oral medication that we have found a compounding pharmacy to provide for her. But actually, you could just go to Amazon or the pharmaceutical store down the street and find it. It's known to be safe, and it actually had been used before. One of the reasons we were able to jump to this really quickly is it has been used before in other biochemical defects upstream in this pathway, and so there was a nice safety history for that compound to be used. And her red cells look better. So are we there yet with precision medicine and precision diagnoses? I think I've mentioned a couple of times along the way, sometimes where it makes a difference in our own cases. I've listed CAD here on this list. There's a whole list of these genes, and I'm happy to share the slides. Obviously, they're being recorded, but I'm happy to share this with anybody who wants it by email if you want to send me an email. But I think increasingly we're finding variants in genes where it does make a difference to know, and we're either initiating trials like the one with the CAD, or we're actually initiating treatments that are pretty simple and straightforward. So for Berg, had we known he had a KCNQ2 variant when he was an infant, we didn't have ezogabine, which is a potassium channel opener yet, but we now know that sodium channel blockers, because of their interactions, things like good old carbamazepine, actually work reasonably well for the seizures in that setting. And so if Berg had shown up today and we saw tonic seizures and a disorganized EEG, we would have done either a rapid exome or a rapid panel. I would have done a rapid panel at my institution, gotten a KCNQ2 variant diagnosis quickly, and put him on carbamazepine, which otherwise we don't tend to use in newborns, right? It's really hard to maintain levels. You've got to dose it multiple times a day. It's not an IV loadable drug, and so it's not one we use in the NICU all the time. But in this setting, I would have done that if I had known the gene and that he had a pathogenic loss of function variant. In point of fact, I found out about Berg's variant long after his seizures stopped. So they now give the parents a community, give us a place to go with natural history studies. Maybe will give us a treatment to try to improve development. But I really wish we would have had that variant back when I was treating his seizures when he was an infant. Again, many of these that I put in red earlier, these are genes that we see. These are genes we see on a regular basis in clinic or that we have referred to us and that are coming out of our pilot studies now. So they're not as rare, I think, as was originally thought. And there are groups of parents joining together with physicians and with researchers to try to understand them better. So with that, I'll stop by saying we're heading in the right direction. We're bringing the basic science together with basic neuroscience. We're figuring out how to do precision treatment either as N of 1 stories. We're being careful with gene therapy and ASO trials that are emerging to do things the right way and to do things ethically and to try to make sure that we make both testing and treatments available to all patients with epilepsy. And we're thinking about what meaningful outcomes really should be, whether they're seizure related or development related or related to something else. So what I think we'll be able to do is not just say we've got epilepsy and it's either solved or unsolved, but we'll be able to say if you're solved, you'll fall into either an mTOR pathway or an ion channel gene pathway or something else. And if you're in one of those pathways, you'll have a corresponding treatment. Whereas if you're unsolved, maybe we can do expression profiling. Let's look at the RNA if the DNA is not giving us the answer and put people into the same categories and offer the same treatments. And of course, it won't just be 3 different types. There's going to be lots of different types, but I think we're on the verge of being able to do that. So that's where I see the field heading. It starts with each of those cases one at a time. It starts with these experiences where we can build our knowledge, and then we'll be able to group patients according to biology and group them for trials and hopefully change their outcomes. So with that, I'll thank you for your attention. I hope there might be a couple of questions, and really, really a pleasure to be able to do this. I wish I could see you and interact as we normally might do in person, but hopefully we'll be able to do that in coming times at the AES meetings.
I have not had any questions come in the chat, but feel free to raise your hand or try to unmute yourself if you have any questions, or add them in chat.
I'm going to stop my share and maybe I'll be able to see you guys. I don't know if people can show themselves on the video. I see some more familiar faces though. I don't want to call you out and embarrass you, but feel free to ask questions. Or make comments. I know some of you have given talks like this in the past, so if you have comments or things you'd want to add or to make sure that everybody else hears, open to that as well. You know who you are, but I won't call you out.
Hi, Anne. This is Louis Dang.
Hi, Louis.
I was wondering about your comment. Sorry, I joined your talk late, so I'm not sure if you covered this, but for those variants that might happen at low frequency in a mosaic manner in a small area in the brain, how do you think we can try to discover which genes are involved in those, or do you think that we'll just kind of infer which genes those are from the larger dysplasias and surgical resections?
I think that's part of it. I know you're doing some really cool work in the area as well. I think part of it is going to be drawing from those malformation genes and saying, well, if we see them in these large visible malformations and then we see them in smaller malformations, at least when the cells look similar, let's look for them in smaller number. But I think we also have to just take a little bit more of a genome-wide view and say, well, if SCN1A, for example, can cause severe epilepsy when it's present in all of the cells, maybe if you've got a handful of cells in a small area with an SCN1A disruption, that can be the answer. Finding those is going to be very hard. I think part of the reason it's going to be hard is that we hypothesize that it's going to be on the order of 1% to 2% or fewer of the cells. And generally speaking, these people aren't necessarily having surgery, but when you do have surgery and a resection, it's a small amount of tissue. So if you don't have a great idea of what you're looking for in terms of a panel of genes, it can be harder to discover that. But I think it's going to be one of those two things, a targeted approach from what we know, but then really an unbiased, untargeted approach.
Great, thank you.
We did have another question come in. It said, could you please comment on the role of genetic testing as individuals with unsolved cases in childhood transition to the adult setting?
That's a great question. So this is something that has been an area of conversation at AES, largely led initially by Sam Berkovic, who's an adult epileptologist involved in a lot of the early epilepsy genetics work, and taken up more recently by Danielle Andrade in Canada and several colleagues. Danielle actually joined the faculty of the epilepsy genetics workshops that are run by AES, in part because we wanted to be able to cover that population. And so I think there's a huge need, and I think what we're seeing now already is that patients who are, let's say, 30 years old, who had childhood onset, let's say, infantile spasms and went on to have Lennox-Gastaut, they show up in an adult clinic as adults with epilepsy and intellectual disability. And of course, you may have that history, but when they were infants, there was no genetic testing. And so they didn't get tested just because of the time artifact, right? So I would say it is worth looking at each adult, just as we need to remind ourselves to look at each child, but particularly in the groups where the yield is high. Those with intellectual disability, those that started with those early onset syndromes that we know we have a targeted hypothesis list that we're going after when we're doing testing. I think those are the cases of that when they're adults, you have to sort of try to figure out what did they look like when they presented, how did this story unfold, and then go from there and do the appropriate testing. It's certainly harder because I think as much as we have not enough infrastructure in the pediatric epilepsy world to do this routinely, there's even less in the adult world. And so I think we've really tried to see if we can tap the next generation and train some people, and ideally train across. We all do some adult neurology when we're pediatric neurologists and vice versa, but we've had fellows come to us, and I think fellows around the country who are adult epilepsy oriented, but perhaps their research is pediatric epilepsy oriented, or perhaps they're interested in adults with neurogenetic syndromes. And I think the more that we can get ourselves into each other's clinics, the better, the more we'll be able to translate this. So I think it needs to be done. There are more and more series coming out. There have been a couple of papers in epilepsy recently, and some attention given to this, because it's got to start with every time you see a person, you go through your list of what are their causes, and you've got to ask yourself, if I don't have a cause, is it something genetic? And then how do I go about doing that? I think first there needs to be a mindset change across the field. Present company obviously are motivated and interested in this, but I think across our whole field of colleagues who are perhaps not even epileptologists, who just need to be reminded that even though 30 years ago we may have learned about idiopathic epilepsy, no cause unknown, now we've got to think of those as potentially explainable. One more question about this is from Geeta, related to a POLG mutation and treating with valproate. This is regarding a couple of patients with intractable epilepsy, improved but to a small extent on valproate. Once genetic testing came back with POLG, Depakote was discontinued even though there were no liver effects seen, and the epilepsy is poorly controlled. So this is an area where there's, I think a little bit of knowledge early on, where there are a couple of variants in POLG that were associated with adverse liver consequences in the presence of valproic acid. That has led people to see the gene POLG and to take people off of Depakote, with good reason, right? You don't want to have a valproate-induced hepatotoxicity if you can avoid it. What I have told colleagues, and we've had many cases come up, that if you've got a patient who is on a stable treatment with valproic acid, and you get this genetic result back, first just pause. The first thing to do is to see if those specific variants in POLG are the same ones that have been associated with the adverse consequences, and sometimes they are, and those are the ones that tend to get reported. But the second thing is to say, well, do they have any other indication of POLG, or is this just a heterozygous single change that's not necessarily likely to be pathogenic? And so really think hard about, does this fit? And then it's a balancing act, and if they're doing well, and if they were previously not well controlled and they're doing well now, typically I think people will err on the side of keeping them on following labs as they normally would. If they weren't doing so poorly, and people sort of have the logic, well, why keep them on something they don't have to be on? And then the situation comes about that they're now doing poorly again. Do you want to try them on valproic acid? I think if nothing else is working, you have to just put the facts out there for the family and say, look, there's this theoretical risk. This is what it's based on, this one study in most cases. Your child or this patient did well on it before with no adverse consequences. We've tried XYZ other options. We're sort of now back to this. If we try this, it's going to be with this type of surveillance and this type of monitoring. I think once you're off and you've got to do an active process to start them on again, it's a little bit more difficult. If you can not stop them, if they're doing fine, it's a little bit easier. But it really requires having an educated discussion with the family and really explaining that it's not a black and white issue that this drug is bad when you've got a variant in this gene, but it's actually quite complicated and there's a lot we don't know. So that's a little bit of a circuitous way of saying it really depends, but I certainly haven't recommended uniformly that people come off in the presence of those POLG variants. I've recommended sort of cautious maintaining and keeping them on, especially if they were previously refractory. I'm curious to know if anybody else out there has other practice in that area. We're sort of using best evidence that we can, which is limited.